Jonny Perl has agreed to join us today. Jonny and I have a serious passion for chromosome mapping, and I'm so proud of him for developing a tool which has made it so much easier for us to map our chromosomes and the other things he's done with his other tools as well. So I very much appreciate everything that Jonny has done for the genetic genealogy community. And without further ado, I'll go ahead and let Jonny maybe give a few other introductory remarks, and he's going to give us an update on DNA Painter and everything else that he's been working on. So go ahead, Jonny.
Great. Well, thank you very much, Tim. And thanks also, Lisa. Thank you both for having me. And thanks everyone for coming along today. So yeah, I wasn't quite sure how to pitch it today because I know this is quite an advanced audience, but I don't like to jump straight into kind of more techie stuff in case I sort of leave people hanging. So hopefully you'll find I've kind of pitched it okay. I'm going to talk a bit about DNA Painter. I'm going to give you a quick reminder of what it is, and then I'm going to try and showcase some recent developments. And then we're hopefully going to have a little bit of time afterwards where I can actually do a bit of hands-on stuff. So if I demonstrate something in the talk and you're wondering, how did he do that actually? You can ask and I will demonstrate it on the screen, for example. I'm going to touch on what the site is and sort of tell you why I built it. Give you an overview of what it does, but also what it doesn't do, because it doesn't try to be a one-stop shop for everything a genealogist might want to do. Then, as Tim touched on, I'm going to jump into chromosome mapping, which Tim correctly identified as one of my passions. And I'm taking an approach to it today where I guess I have a really quick recap of what it is, but then I dive into segment interpretation and how I think in a way chromosome mapping gets a bit of a bad rap, particularly at the moment with the kind of segment crisis where 23andMe have removed segments and MyHeritage have removed bulk segments. And a lot of people obviously say, well, you don't really need segments because for the thing they're doing, you don't really need segments, but it's not to say you can't do really cool things with segments. So I'm going to hopefully get that across, and I'll touch on a few future development plans. And then I will talk about the other big reason, I guess, that people use DNA Painter, which is the relationship prediction tools. So I'm talking about the shared CM tool with its histograms, another version where you can put two amounts in, and then also what are the odds and the new plus version of that. So very briefly, I'm a genealogist and a web developer. I'm based in Swansea in Wales, which doesn't look anything like that today. It's a slightly dismal day, although not too bad. We had a bad storm yesterday, but it's down on the southwest of Wales. I guess I'm a relative newcomer to all this. I think a lot of people here are much more experienced than I am, to be honest. I only tested first in 2016. You could say I've tried to make up for it since then by thinking about it and working on it quite a lot. And as Tim noted, I founded a website called DNA Painter, and that was back in 2017. So it's a website, and the reason I made it, to be honest, was I just got my results. I was trying to learn what they meant, and it occurred to me that given that I was a web developer, I could try and make a platform where other people could also learn more about it. And that's really all I had in mind, first of all. But then I was lucky enough to meet some quite interesting people. The site's evolved, and at this point, it hosts quite a lot of different tools that I hope will be helpful to anyone, I guess, who's a genealogist working with DNA. Because let's face it, we've all got these huge match lists, haven't we? And we would all love to know how these people on a match list are related to us. And there are lots of different approaches and techniques that can help us with this, aren't there? Obviously, you can look at shared matches and you can try and build genetic networks consisting of groups of matches who are related to you via different ancestors. And you can even partially automate that process. So you've got these things called auto clusters, MyHeritage Genetic Affairs at GEDmatch. You can also get them via DNA GEDcom. And then we've got the segment-based approach, which you might also like to try, and obviously I advocate for that quite strongly. To be clear, it's not like I think that's better than anything else. I am an omnivore for all, I do all these things. I'm not, it's not as if I just use my stuff. In terms of the relationship prediction, obviously that's really useful if you've done that work to make these genetic networks. You can then use probability tools to look at different ways that these networks might connect to you. So that can be quite powerful. But as you'll know, if you do this at all, none of these things replace the genealogy. And in fact, probably the main thing that all these techniques do is they give you new genealogy to do by creating new potential research avenues. So if you don't like genealogy, obviously you're in the wrong place. As far as I'm concerned, the reason I don't try and do clustering is because someone else has done it. So I figure I may as well focus on the things that haven't been done really well by other people. So that's why there's no clustering tool, for example, at DNA Painter. There are a lot of different tools that I probably won't have time to talk about, but on the tools page, you can actually find a listing of them, and there's generally a blog post explaining what the tools are and what they do. So today, I'm going to focus on a kind of subset of those tools. Just to be really clear, in case there's anyone on this call who doesn't understand this, you don't upload your raw DNA data file to DNA Painter. So data that the site does work with consists of segment coordinates, so the chromosome number, the start position, the end position, the number of centimorgans shared, and that kind of thing. And then separately, just alone, even if you don't have the segment data, you can use the probability tools if you just know how much DNA you share with someone in centimorgans. And then finally, you can also load GEDCOM files into various tools on the website, so you can extract portions of family trees, essentially, so that you can work with them. So chromosome mapping. Like I said, I'm going to remind you what it is. Probably most of the people on this call know what it is, but I'll remind you quickly what it is. And then I'm going to show you a practical segment analysis technique that I use. I've been thinking about for many years, but it's kind of all crystallized recently when I was able to get some empirical data that kind of proved it, that was right, that I wasn't crazy. If I go too quickly over something that you don't understand, there I do have a more detailed introduction about chromosome mapping, which I did at Family Tree webinars, and I've made a little link to that you'll find at the bottom of the next few slides. So chromosome mapping, in a nutshell, we've got our 23 pairs of chromosomes. Ignore the fact that I haven't dealt with the X and the Y here. Let's assume this is a female who's got two X chromosomes, and one copy of each chromosome comes from our father and the other comes from our mother. So far, so simple, right? 50% from each parent. But then within these chromosomes, we received different proportions kind of randomly from each grandparent. And that's where inheritance kind of gets going and has fun. You may find that you inherited much more from one grandparent on one side than the other. So this is my current chromosome map. And the key on the right gives you a sense of what some of those colors mean. So the yellow is DNA I've been able to assign to my paternal grandfather, the blue to my paternal grandmother, etc. And just to be completely clear, none of my grandparents have taken a DNA test themselves. I'm 50 years old and my grandparents died quite some time ago, unfortunately. So normally when you map a segment, it's not based on the actual people. It's not your ancestors who've tested. It's their descendants who've tested. And you've then made the assumption that DNA, since you share it with this person, is likely to have come from that place. So if we run through a really simple example, I might have a third cousin on my match list, and my match list might say I share 128 centimorgans of DNA with them. But with chromosome mapping, it's not just about those 128 centimorgans, which indeed is a totally reasonable amount to share with a third cousin. It's also about the specific locations where we share that DNA. Now, annoyingly, as I'm sure most of you know, you can't get this information from Ancestry DNA or anymore 23andMe. But on the flip side, you can get it from Family Tree DNA, you can get it from GEDmatch, from living DNA, and also from MyHeritage. Essentially, the process of mapping these chromosomes is, okay, this is my third cousin. If we share DNA, where is it likely to have come from? So it's based on the assumption that the DNA you share is based on this known relationship. So in my case, we can see that we share a set of second great grandparents, and that's Charles Jones and Mary Moore. And if I map that and I have a look at it in the context of my entire map, I can see if that makes sense. So, for example, do those segments sit within the correct grandparent, for example, or do they not? That can help me check it. Because what I want to know is, did all this DNA definitely come from Jones and Moore? Now, the most likely explanation is, sure, why not? He's my third cousin. I know exactly how I'm related to him, and we share DNA. Now, just me and you, or just someone on the end of the line, we're unlikely to share DNA by chance, but it does happen. It's possible that Mike might also be a really distant cousin on another branch. He might be my sixth cousin once removed. Obviously, if you have endogamy or pedigree collapse in your tree, you'll be familiar with this challenge. Sometimes not all the DNA you share with someone is from the relationship that you think it's from. And you don't even have to have pedigree collapse or endogamy to have this experience. I've got a lot of people, particularly in my wife's family, who have history in very rural Oxfordshire. And I think some of those bits of DNA are just knocking around in the population. So they're the types of segments, I guess, which Ancestry DNA sometimes would strip out as part of its timber algorithm. Kind of have to watch out for those. And obviously, the more distant a cousin is, the more likely it is that actually that DNA isn't from the relationship you know about, but from another one. Because, maybe they're your seventh cousin, but they're also your eighth cousin or ninth cousin some other way. Also, some of the segments could be false positives. So inevitably, if you're mapping your chromosomes, it's quite a handy way to see that because you're seeing the segments in the context of the other DNA that you've mapped, you can see if something's obviously wrong quite easily. Another thing I'll touch on is these nice, precise-looking numbers that we get, of course, are not really precise necessarily. They're based on SNP testing, so we're not looking at whole genome sequencing at this point. So that means that the start and the end points aren't necessarily going to be the true start and end points. And we'll see some examples of that later. But yeah, it's a work in progress, my map. It's never done. I've done a lot of work on it, as you can see, but it's never done. It's somewhere where I can write some notes, I can identify inconsistencies, and I can also identify otherwise unknown matches. So for example, if I've got a match who's only at family tree DNA, I can potentially compare them to other people who've tested at MyHeritage. And so I can't directly compare them each other on the testing site, but if I've got the segments that I share within my chromosome map at DNA Painter, and I know, for example, they're both paternal, then I can see if they're likely to be on the same line or not. Ultimately, for me, I think the map it's a concise DNA research log, right? And just recently, that's become really, really valuable. We all know people delete their DNA test results sometimes, or you can have situations like 23andMe where suddenly those segments are just removed. And I've had loads of people write to me and say, wow, I'm so glad that I imported this data into DNA Painter because now I haven't lost that segment data. So it's useful to kind of put everything together in one place where you can easily find it. And a recent development means that this isn't actually just limited to autosomal DNA. So as of a few months ago, you can also add Y and mitochondrial haplogroup information to your chromosome map. And you can even assign them. So you can assign them to the furthest back ancestor on that line. So my Y DNA test, I've done the big Y700 at Family Tree DNA, and we can see my very specific haplogroup I've been given. And I've assigned that to a man called Isaac Perl, who was born around 1800, either in Bohemia or in kind of Lower Silesia, so around the Czech and Polish border. So that, as a kind of completist, I find that quite pleasing that I can put my furthest back maternal and paternal ancestors, so matrilineal and patrilineal ancestors into my map. And another feature of that is that you can actually explore that information. So it'll automatically generate links out to scaledinnovation.com. So I know Rob Spencer appeared here a few months ago and showed his amazing website, which has got maps of kind of Y-DNA migration paths. So you can link straight into that for your haplogroup. You can link over to the family tree DNA resources and also link up with mitoY DNA to compare with other people. So if you share your map with someone else, they could click and compare themselves to it. I like to think of a chromosome map as another slice of your research, if you like. It's a companion to your direct line. And you can actually, I'm not going to touch on it too much today, but you can actually import your direct line to DNA Painter as well in something that I call an ancestral tree. As of quite recently, you can upload pictures for each of your ancestors there as well. So essentially, you select your GEDCOM file, you select yourself from that file, and then it'll extract your direct line. And you can annotate it, you can use different color palettes, and you can put pictures in. Now, again, you might think, why would I bother doing that? I've got my tree in a million places. The main reason to do it is that you can share it and it's then searchable. So if you've got a particular ancestral place deep in your tree, if you've shared your tree in this format with a DNA match, it will hide all living people by default, as we can see on the screen there, but they can actually search on ancestral places and they will come up. Another nice thing about this is you can explore inheritance paths actually overlaid onto your own map. So this is my great-grandfather, James Brown Matthews, who I don't know if I'm particularly interested in because he kind of looks a bit like me. But anyway, theoretically, he is the source of 25% of my XDNA. And that is quite interesting to me because, in fact, I did inherit most of my XDNA from my mother's mother. So it is on that side, a lot of it. So that gives me that connection, lets me understand that inheritance path as overlaid on my own genealogy. I've been talking about this for a few years and I haven't quite done it, but I'm definitely getting closer. In future, you will actually be able to link your ancestral tree to your map. So at the moment, when you have your chromosome map and you map some segments, if I wanted to map, for example, to Charles Jones and Mary Moore, I would just have to type in Charles Jones and Mary Moore or select them from a drop down where I've typed it in before. So in future, you'll link your tree to your map, and then you'll be able to select from an ancestor. And obviously that potentially opens up lots of very interesting display possibilities in terms of inheritance and which line did this segment pass through. And I guess it isn't the most exciting part of it, but one thing I'd like to be able to do is a display a little bit like this one, where you can actually display a picture of your ancestor on your map next to the segment that you inherited from them. So that'll feel like a milestone when I do that, even though it's not really, strictly speaking, of scientific value. Now, I'm going to show you a few ways that I use chromosome maps now. What I want to emphasize is that I'm not trying to be super prescriptive. What I've built here is what I hope is quite a flexible piece of software. And really, you can do what you want with it. A lot of people have used it in ways that I've never really thought about. For example, people have done descendancy studies, all kinds of stuff. But I'll just going to show you how I've been using it, and hopefully that will spark some ideas. So very, very briefly, when you map a match, you need the segment data. I've got a help page that shows you where you can get that. I'm guessing most people on this call know where you can get it anyway. There are some videos even showing you where you get it from the different websites. And here is how you would map a match from MyHeritage. So within the match detail page, you click download shared DNA info. MyHeritage give you a CSV file, and you have to open that and then paste the data into the Painter match form. And then you hit save match now. And then you have to do this process of thinking, well, who are the common ancestors? Who did I get it from? And in this example, I've got someone called James, who is the brother of my previous example, Mike. So again, the common ancestors are Charles Jones and Mary Moore, and I can enter their names in and I can hit save match. And if that seems incredibly cumbersome, you can do bulk import. It's just at this point, it's only GEDmatch and Family Tree DNA who provide these bulk files. It can be really interesting to do that, particularly at GEDmatch. If you have a phased kit at GEDmatch, if you're lucky enough to have a parent tested, then you can do a segment search on your phased matches from each side, and then you can in bulk import maternal matches and paternal matches separately. So that I do find valuable. It's just if you just have a morass of segments and you're not sure if they're maternal or paternal, that can get messier. Like I said earlier, DNA Painter Map as a kind of repository for data which you don't know what the future accessibility of is, I think is quite a powerful thing. Just to give you an overview of what's happening with segment data, basically in October of 2023, a few people who reused their passwords unfortunately led to this regrettable situation with 23andMe, where someone rather deviously got lots of data off and then posted about it in a kind of braggart way, and then things took off from there. And what that means is that at this point, you can't get segment data at all from 23andMe. Obviously, you could never get it from Ancestral DNA anyway, but you can still get it from Family Tree DNA, GEDmatch, Living DNA, and MyHeritage. And then in terms of the bulk segment data, you can't get bulk data from Living DNA, you never could, but MyHeritage removed that feature around that time. I don't know if it was connected or not, but just around late 2023, they removed the feature where you could download all of the segments for all of your matches. Family Tree DNA actually did the same, but luckily they brought it back, and GEDmatch remains a place where you can get bulk segments. And then the other type of segment that you can map, which is quite interesting if you're a completist like me, is ancestral population segments. You can actually get these, weirdly, from Ancestry and 23andMe, the sites where you can't get any other segments. 23andMe still allow you to download those population segments, and Ancestry give you a picture of the segments, and I've actually got a special tool where if you copy and paste what's on that page, it will generate the segments for you and you can use them in your map. And then from Family Tree DNA, you can download the segments and import them via the import segment data tool within DNA Painter. In terms of interpreting these segments, we talked about what they are. I guess what came to me, and this was a bit of a slow burner for me over the last few years, how can we learn as much as possible from these segments that we've gone to all this hassle to get? How can we figure out with as much specificity as possible where they came from? And I don't know why I have this burning desire to do that, but I do. What do I get out of it? I guess I get to feel like, okay, well, I really did inherit this bit or this much from this ancestor. And potentially it means that if I've got unknown ancestors, I can pin down which branch they're likely to be on with more specificity than maybe I first thought that I could. As you'll gather, this is something I've spent a bit of time thinking about. Let's consider Steve. Steve is my maternal first cousin. We're looking at all of the segments that I share with them. Obviously, that's quite a lot of DNA one can share with a maternal first cousin. And so what can I say about these segments? When I first started out with chromosome mapping, I was really, really cautious. I think because I didn't know that much about it and I didn't want to be making assumptions that I couldn't really make. So I started off thinking, okay, well, if I've got a first cousin, what do I know about it? All I know really is that these segments have to be on my mother's side because Steve is the son of my mother's sister. And I just kind of left it there. My first iteration of chromosome mapping, that's what I did. I just used them as an extra way to phase matches coming in. Okay, well, they match him or they don't match him, so they must be on maternal side or paternal side. Then I started to think, well, wait a minute, maybe I should think of it as my maternal grandparents. It could be one or the other, but maybe not both. I think I did this on the basis of someone else saying it rather than really having thought it through. So then I thought it through, and here is the way I think it all happens, and certainly all my research so far appears to back this up. For me and Steve, as first cousins, our common ancestors are our grandparents. So that's Joseph Hill and Anne Matthews. And we can see there under the orange arrow an example chromosome of what they inherited. So on Joseph's paternal chromosome, he inherited whatever DNA that Sam Hill happened to give him. And on his maternal chromosome, he inherited whatever Agnes Dunlop happened to give him. And for Anne Matthews, the same applies for her parents, James Matthews and Rhea Marshall. These coloured strips we can see, those are effectively the reference chromosomes. Those are all the different bits of DNA that me and Steve could possibly share, have to be taken from there. When my mother and her sister June were born, as siblings, they share, I guess, about 50% of their DNA. And my mother's ancestral segments look a little bit like this. So if we zoom into this and have a bit of a closer look, we can see that on her paternal copy of chromosome 1, she's got a little bit at the beginning from Agnes Dunlop, and then she's got the blue bit from Sam Hill, and then she's got a crossover and she's got more from Agnes Dunlop, then back to Sam Hill, then back to Agnes Dunlop. And then on her maternal chromosome, she's got a bit from Ray Marshall, then she's got effectively flipping back between the two of them again, as we'd expect, as we've all seen in our own results. Then for her sister, obviously it's a different blend of segments. And if we have a look at where they match each other, it's complicated, isn't it, with full siblings, because you've got a mix of half identical and fully identical regions. It's a bit strange when you look at it. Now, one of the brilliant things that happen when you have children is that you can't pass down a fully identical region, certainly not very often. It's, not unless you've got some kind of pedigree collapse. Once me and Steve came along, it's all just on the maternal copies of our chromosomes. So in any particular position, we can only have inherited DNA from one of those great-grandparents. That's kind of powerful if you actually think about it. If we now look at the segments that me and Steve share, we can see something, can't we? We can see that each matching segment is actually from just one of those great-grandparents. When I thought this through, I thought, well, that's quite cool. If we then look back at the first cousin segments that I share with Steve, it's actually something like this. It's not just that, these are from my mother's side. It's not even just these are my mother's parents. It's actually these are from a specific grandparent of my mother. And so from that, I kind of extrapolated a rule, and I'll talk a bit more about this, but fundamentally, each segment that you share with a cousin will have been inherited from just one of the common ancestors' parents. So in this case, it's one of these great-grandparents. And to be honest, it kind of seemed a bit too neat and tidy for me when I first came upon this. And so I wanted to prove it to myself that this was really real. And as you can imagine, it took a bit of time to get the data to do that. And it was just last year that I managed to do that. I've got two great nibblings, as we would call them in this country. I've got a great niece called Flo, and I've got a great nephew called Nick. The amazing thing about Flo and Nick, who are first cousins, is that 3 of their 4 great-grandparents on that side have actually tested. And the nifty thing is that the one who hasn't tested, I can actually just infer the segments that they would have got from him, because it's all the ones that are left that they don't share with the other great-grandparent who has tested. That means the process of making a chromosome map for these people is incredibly quick. I don't have to mess around with matches and think, who's the connection. I can literally just directly compare Flo, in this case, with each of her great-grandparents, apart from the one who hasn't tested, and then I can infer the rest of it. So I was able to make this comprehensive maternal chromosome map for Flo, which was really cool. And then I had a look at the segments that Flo and Nick share according to my heritage. And this is them. There are 38 of them. And if I overlay those on top of the map, obviously what I'm hoping is that each of these yellow segments are going to line up with just one of the 4 colours representing each of the great-grandparents. I had to check the source of each segment quite carefully. Within DNA Painter, you can click on any chromosome number and it will expand it, so you get this kind of stacked view with the segments. And the brilliant news is, yes, it does work. We can see that all of the yellow segments can be associated with just one great-grandparent. Now, it's worth recapping that, the ends of the segments are fuzzy. And I guess if you're new to DNA, you might say, well, no, that segment with Nick is a tiny bit of it's from CW and the rest of it's from BB. But obviously, I've been around the block long enough to know that the segment, the start and the end point are fuzzy, and I don't actually have to worry about that. So that, I guess, filled me with excitement in a way to think about how I might be able to apply this to my own research. I should briefly just show you a couple of other features. So within DNA Painter, there's a settings panel at the top, and we'll get into this a bit more in the demo afterwards perhaps, but a few options that are really handy if you're in this kind of view are to show the centromeres on the chromosomes, to show the megabase guides on the expanded chromosomes, and then to show the key line. So you see the line there with the 27 million number next to it. That's following my mouse around as I move it around the screen. So if I'm just trying to figure out, well, what is the overlap there? Do I have to worry about that overlap? Or is it likely just a kind of fuzzy segment thing? That allows me to do that. And there's also a slider at the top. So if you've got, as I do, a very, very full chromosome map, and you've imported all kinds of segments into it, you can actually adjust the displays. You can say, actually, I just want to see the segments that are 20 centimorgans or larger. And this is a new feature that came in, I think last year or maybe at the end of the year before, but it's one that perhaps not everyone's seen. To get back to the topic at hand, we went from these segments that Flo and Nick share, that previously they might have just said, okay, well, these obviously came from my grandfather on that side, to actually, look at this, we've actually been able to assign them all specifically to a great-grandparent. It wasn't completely plain sailing. There were two problem segments, and I guess that probably won't surprise some of you. We know that sometimes, particularly on certain sites, segments can be fuzzier than on other websites. And what I found was that for this segment on chromosome 7 and the segment on chromosome 19, they'd be merged. MyHeritage had merged them into one. On chromosome 19, it was kind of understandable. On chromosome 7, it was a bit strange because there was quite a big gap. And in order to fix that, I had to go to GEDmatch to get a second opinion. And then I also had the parent kits for reference, so I could actually see what parents of Flo and Nick shared with those great-grandparents of Flo and Nick as well. Do I have to worry about that? Not exactly, but I guess it's something to keep in your head as a caveat. 4 of the 38 segments that were reported by the testing company had actually been merged. So if you were investigating a segment yourself and you thought, well, it doesn't make sense for this to just come from one ancestor, that's a possible explanation. Yeah, very nice for Flo and Nick, right? Good for them. They've got 3 great-grandparents tested. But for the rest of us, obviously, we might not even have parents tested, let alone grandparents or great-grandparents. So it's not as straightforward for us, is it, to classify these segments? So I thought I'd do a more realistic example. Lorna and Tim are also first cousins, just like Flo and Nick, but unlike Flo and Nick, none of their ancestors are actually able to take a DNA test. So it's just Laura and Tim. That's what they've got to work with. Now, they share 27 segments of DNA. And so based on the rule that we've been talking about, each one of those segments should have come from just one of these 4 great-grandparents. We've got HC, MD, FB, and FG. So if you don't have the luxury of testing a great-grandparent, you actually just have to think about, well, who else does share DNA in these places? And then who are they? Can we link them exclusively to just one of these 4 great-grandparents? What can we do? Now, ideally, you're going to find someone who is an actual identified cousin, like a 4th cousin. That'd be brilliant, wouldn't it? Because you'd feel really confident. But, well, A, that's probably unrealistic sometimes, depending on your matches. And B, it doesn't necessarily matter. In my ancestral background, quite often if I don't know who someone is, I'll know which grandparent line they're on, for example, either because of their ancestral population, their ancestral background, or because of other shared matches that they have. Now, in this case, we've got a 3rd cousin that we've identified, and that cousin is definitely descended from a sister of this guy, FB. So what that means is we're onto something here. We can start to classify the segments. We can see that Claire shared 82.5 centimorgans with Lorna, and so conveniently MyHeritage has this triangulation tool. So if we use that in their chromosome browser, we can see that there are 2 segments where Lorna and Tim share DNA and also Lorna and Claire share DNA. So that means I can label those segments now. I can say, okay, well, of the 4 great-grandparents, these ones seem to have come from Frank B. And clearly it was a bit of a journey to get there, wasn't it? So you're going to want to make a note of it in your notes. You're going to want to specify why you've made the assignment that you've made, if you like. But I think with a bit of work, it is possible. For that example, it's my mother-in-law, and I'll admit I haven't identified all 27 of those segments. That's maybe a bit optimistic, but I can start to narrow it down. Sometimes I can say, well, it was either this great-grandparent or this one. It definitely wasn't this one or this one. But you can potentially use inferred matching if you've got siblings you've tested to try and narrow things down more. It's all part of of the joy of the hunt, if you like. So let's try applying this rule to some other relatives just so I can talk about how it works. So I've got this guy, Chris. He is my father's first cousin. So to me, he is an older generation first cousin once removed. In this case, I just want to find the common ancestors. So that's his grandparents in this case. And then each of the segments which I share with him is going to be from just one of their parents. So one of these 4 people that we can see on the screen. If we take another example, this isn't even a cousin relationship, it's a half relationship. If it was a full uncle, I couldn't really use this rule because some of the segments I share with a full uncle are going to potentially cross over both of their chromosomes. But because it's a half uncle, we've just got one common ancestor who is my grandmother. And so any segment I share with my half uncle would have to have come from just one of her parents, according to this rule. And if we just return to the first match I talked about, you may remember that I have a third cousin, and the common ancestors were Charles Richard Jones and Mary Jane Moore. And we looked at 5 or 6 segments that I shared with that cousin. So it's not just Charles Richard Jones and Mary Jane Moore that I can assign them to. Potentially, I can assign each one of those segments individually to one of the 4 parents of Charles Richard Jones and Mary Moore. So I've got Richard Jones, Anne Maria Hayington, James Moore, and Julia Blackburn. And that's actually kind of exciting. These are people born almost I think Richard Jones was born in the 18th century and the other ones were born in the very, very early 19th century. So I'm reaching back quite far in time, which is an exciting thing to do, actually, with DNA and with segments. So I extrapolated the rule into a simple one, which is that each segment you share with an nth cousin will have been inherited from just one of your nth great-grandparents. So if it's a first cousin, then it'll be just, from one of your first great-grandparents. But if it was a fourth cousin, with the caveat that obviously you're not quite sure about these segments that you share with people too far back. If it was a fourth cousin, then it would be from just one of your 4th great-grandparents that you have in common with them. And if it's a removed cousin, then you have to use the older person's nth great-grandparents. Okay, I hope that made sense. So when doesn't this work? I touched on this before. Effectively, it doesn't work for full siblings, kind of for obvious reasons. We share some fully identical regions with our siblings, so we can't talk about segments in quite the same way. Direct ancestors, it doesn't work. Why doesn't it work? Because a segment that you share with a direct ancestor might actually cross both their copies of the chromosome. So again, you can't say the same thing. And it also doesn't work for full siblings of ancestors for the same reason. So that's why a full uncle or aunt wouldn't work. But a half uncle or aunt did work. Also, if you have near identical crossovers, which I guess is probably what happened in at least one of those 2 cases with my real life data, possible potentially for the testing company to say, okay, well, if these people have crossovers in exactly the same place, maybe it's beneath the margin of error for a match, and they might actually merge these segments. So that purple and green segment might actually be merged into one. You won't be at all surprised to hear that if you've got pedigree collapse, it's going to mean you can't actually narrow down the choices quite so much. And it's probably going to get very confusing. If you've got endogamy, theoretically, there isn't any reason that the rule can't work. It's just that you're going to want to use a larger segment threshold, which you'll be used to doing anyway if you have endogamy. So that is today's whistle-stop tour of DNA Painter chromosome maps. Now, I still really enjoy working on mine. I mean, it's been 7 years and I still get a kick out of it because people do still turn up. Like a new second cousin to my father turned up the other day. And so that's a lot of interesting segments for me to work with and, potentially make those colors more granular and fill in more detail. So we're going to talk now about the relationship prediction tools, DNA Painter, and the latest on those. And what they're for is really the bread and butter tasks that we face as genealogists working with DNA. A lot of people, their complaint about chromosome mapping is, I think they struggle to connect it to what they're dealing with day by day, which is they just want to know who the missing ancestors are immediately, or they want to help someone find an unknown parent. So every day, the kind of bread and butter tasks are figuring out how DNA matches connect, making a family tree for those matches, and then trying to fit the tester into the tree. That's how I would sum it up. And these are things that are important for any genealogist working with DNA. Effectively, that's kind of the process for people working on unknown parentage cases, even for people working on forensic genealogy cases, or also for those of us working on genealogy brick walls. It's just that for genealogy brick walls, if they're very far back, it becomes harder and harder to do it. But fundamentally, that is the process that we're following. And obviously, the amount of DNA that you share with an unknown match can help you figure out how closely you might be related to that person. And the Shared CM tool is the most visited page at the website, I guess because that's the most widespread question that people have. They will have a match and they'll share 100 centimorgans with them, and they want to know what that means. They might be completely new to DNA, they might not know what centimorgan is, or they might be a seasoned genealogist who just isn't as boring as me and hasn't stored all this stuff in their head, and that's totally fine. Obviously, the testing company estimates have massively improved, and you can probably learn quite a lot from the testing company, but this gives you a fresh perspective. I guess the main thing it does is it helps you figure out how far back you might have to go to find the common ancestor. The tool, as you probably know, is based on this rather amazing data set which Blaine Bettinger has been gathering for, I think, 10 years now. I think 2015 was when he started. So he's made this grid of relationships, and in case you've never looked this closely, within each box you have a relationship name. So here we've got second cousin. Then there's the average centimorgans of DNA, which is shared between those with this relationship. So for a second cousin, the average reported number of centimorgans was 229. And then below, you've got the reported range within the kind of 99th percentile. So for a second cousin, the lowest amount was 41 and the highest was 592. So all my tool is basically that data, except you can enter the amount in. So if you have a match and you share 178 centimorgans with them, you can enter that in, and then it's going to filter the chart below and show just the relationships that are possible for that amount of DNA shared. So to be honest, that might be all you need for now, but if you want to delve in more deeply, you can actually click on any relationship box, or if the relationship is mentioned at the top, and then it's going to open a histogram. And that can be quite useful. It helps you see, particularly if you've got something that's a bit of an outlier, or if you're at the top or the bottom end of the amount you might share, it helps you figure out where it actually sits with the other reports for that relationship. So we can see that for the relationship second cousin, 178 centimorgans is perhaps a little bit lower than average, but it's kind of comfortably in the middle. It's not something that would cause you to worry. I guess if you had a really high sharing amount there and it was just about on the graph, you might think, okay, well, maybe there's a second relationship here. If you had a really low amount, you might think, well, are they a half second cousin? It helps give you a little bit more food for thought. And there is, of course, more. After you enter the number, as well as filtering the grid, the tool's going to give you probabilities for each group of relationships. Like I said, if you click on any of these relationships here, that will actually bring up that same histogram. Obviously, you're going to want to be reasonably up on cousin relationships to be able to understand what all these terms mean, but I hope you are. If you're not, there's a link actually that I think to the Family Search site, which explains what a half second cousin once removed is. And there's actually a new-ish version of this tool I wanted to showcase for you, which is an option to add a second amount. So this is for a case where you might have a sibling who's tested, or you might have a cousin who's tested, like a cousin who's at the same distance away from any mystery match as you, for example. So let's just say we've got a mystery match and I share 80 centimorgans with them. And I think, wow, I wonder what my cousin Mike, my third cousin, shares. I'm going to go on Ancestry Pro Tools, or I'm going to phone him up and ask him. And he might say he shares 200 centimorgans with them. So I can actually enter that in, and because we're the same distance from each other, potentially that means we're the same distance from the match. And so what it does is it filters the grid just like the single version does, but it considers only relationships which work for both of the amounts that you've given. And then if you click it, it'll show both the amounts in the histogram as well, which can be quite useful. So even if you've got like 4 or 5 siblings you've tested, you can just use the lowest and the highest amount, for example, and that helps you to narrow things down. I don't actually picture it here, but just to throw it out there, the other brand new thing in the Shared Centimorgan tool is that it now exists in different languages apart from English. So from memory, what we've got at the moment are Spanish, Portuguese, Czech, Slovak, and Hebrew. Okay, and I've got Norwegian as well. I just haven't quite published it yet. So a slightly unusual selection of languages, self-selected by the really kind people who decided to help me translate 208 different genealogical terms, but hopefully more to come. So if your first language isn't English and the tool annoys you, it'll hopefully be coming in your language quite soon. So we'll move on to what are the odds? Now, what are the odds is really a development in my head of the idea of using the probabilities, but it lets you use genealogy as well as the shared amounts and probabilities. So what are the odds is often shortened to WATO. So I might use that today. If I say WATO, you'll know I'm talking about what are the odds. There's a couple of kind of common scenarios for people using this tool. The first one is you have an unknown match and your goal is to figure out how they connect to your known family tree. And then the other one is you have an unknown ancestor. So that could be an unknown parent, or it could be an unknown grandparent, great-grandparent, perhaps even more distant. And your goal, therefore, is to figure out how you connect to your DNA matches, specifically to the known tree of your DNA matches. Hopefully, you'll be someone who's identified a bunch of matches where you're related to them via this mystery ancestor, and you've been able to join those people together. And then the tool will help you figure out how you connect to their tree. The tool takes the probabilities and it lets you use them alongside a known family tree. And what we're looking at here is the first version of What Are the Odds? Is it the first version? I think it's like 1.5, if you like. It's the version I first launched in 2018, and then I kind of tweaked a few things and added the GEDCOM import capability in 2020. But one of the problems with What Are the Odds is that even though it's incredibly powerful, it's remarkably hard to teach. I don't know why this is. It just is confusing. And I know this from personal experience because when Leah Larkin, whose idea the tool was in the first place, told me about it, it took me about 2 months of communication for me to really understand what she was talking about. So I've got enormous sympathy with people who've always found this confusing. And to prove that, I actually built another version of it, which is a bit easier to use, and I called that WATO Plus. And an extra bonus of WATO Plus is that it lets you factor in the age of the unknown parent. So it will consider all these potential parents and how likely was someone to have been a parent if they were that age in that year. I think that's going to be quite powerful. At the moment, I've only got data from the US, but I have got hold of data for other countries and I'm going to get that in there. I'm hoping that in the longer term, that'll be quite a powerful part of this. So just to recap, the goal is to use DNA to figure out how someone connects to a family tree. And that tree could be your tree. It could be that you've got an unknown match you want to fit into your tree, or it could be a tree that you've made connecting the DNA matches and you want to figure out how you connect to it. To make a really important point before I start, which I think passes some people by because they hear people raving about this tool and they assume that it will do more than it can really do. WATO won't do the genealogy for you. Yeah, you still have to do the genealogy. And actually, the more of the genealogy you can do before you start using it, the better it's going to be for you, the smoother it's going to go. The tool is, I think of it a bit like a jigsaw. Yeah. And if you've got reasonably big matches like 327, 449 centimorgans, it's quite easy. It gives you quite a good picture of what it's like overall. Whereas if you've got smaller matches, like say 45 centimorgans, they can help, but they can give you a misleading picture overall. They can lead you astray. So people always say, oh, well, this is all I've got. Can I use it? And I say, well, yeah, of course you can use it. You can use whatever you like, but it is going to be harder. Useful things to do before you use What Are the Odds? The big one for me is, to actually really work on tracing those family lines down so that you can identify as many potential candidates as possible. So, for example, if you've got a common ancestral couple and you know that they're the ancestors of all these matches, just call them John and Mary, and you're looking for someone who could have been the father of a child who was born in 1950, then it's kind of obvious, isn't it? You want to trace those lines down and get as many males who were born in good time to have been a father of someone born in 1950 into the tree before you start. Because otherwise, all that WATO is going to be able to do is say, well, it could have been someone on this branch of the tree, maybe. And if they were born in this year, then it would be quite likely. You can see how that isn't that helpful. It's not unhelpful, but if you've got actual people, it can be more helpful. And the second thing is to add as many spouses and birth and death dates as possible. Okay, so birth and death dates is probably self-explanatory. If someone was dead, then they can't have fathered a child, for example, so get that information in there. But spouses is actually really important because if you're trying to figure out which of a line of brothers might have been, say, the grandfather of the person who has the unknown mother or father, it helps to have the spouse name there so you can start looking for evidence of that spouse within the matches of the DNA tester as well. So I always recommend working really hard within your family tree software first and filling out the tree as much as possible before you get it into What Are the Odds? Okay, so hopefully that wasn't too abstract because I haven't done the example yet, but here's the example. So this new version was launched in 2024. The old version is still available if you prefer it for any reason, and also because this new version, you actually only get one of them without being a subscriber. So if you don't want to subscribe, then the old version is obviously around as well. So in this example, we can see a structured question here. So within What Are the Odds Plus, as opposed to just randomly saying, oh, this is what I'm trying to do at the top, which is what you did in the old version, you're very, very specific. And I'll show you the specific questions it asks in a minute. So in this example mystery, we have David. And David's got an unknown grandfather. His mother, Kathy, doesn't know who her father was. And we can see that the unknown father is coded in red there. And David has researched his matches. I don't want to understate this part because it's obviously a huge undertaking, but David's a clever guy and he's lucky. He's got these 2 quite big matches and he's been able to figure out how they are related to each other. So he has the germ of a family tree, which potentially is part of his family tree. And so he's ready to use this tool to try and link himself to that tree. And this is the information that he needs to do it. So first of all, you need to have the name and the birth year of someone who's got an unknown parent. Now, in What Are the Odds Plus, that doesn't have to be the person who took the DNA test. It could be your grandparent or it could be your great-grandparent. In this case, it's his mother. So it's his mother's parent. And specifically, it's his mother's father. So we're going to select father, and then we're going to put Kathy in there. Then you need to say when she was born. So we've entered 1944, because that's when Kathy was born. So we're looking, you see how we've given the tool a lot of information there. We said, all right, we're looking for a male person who could have fathered a child in 1944. And then finally, you just have to say whose DNA matches you're using. So in this case, Kathy's not available. So we're going to select Kathy's child, which is what David is. And we're going to put his name in. So then we get this nice structured question that I showed you just now. And next, he's going to import this tree. And as I said, sometimes this tree is going to fall into your lap and sometimes you're going to have to sweat blood to get it. But if you want to solve the problem, you have to figure out the tree, basically, for the genetic tree so that you can figure yourself into it and figure out how you connect in a genealogical way. So he adds the amounts of DNA that he shares with the matches. And then in this example, I'm using an option which is stop WATO from adding unknown people to the tree. This is another innovation of the new version. In the old version, a classic kind of car crash scenario would be someone would add a few matches in, 30 some centimorgans here, 50 here, and then they'd hit suggest hypotheses, and their computer would lock up for a few minutes, and then it would give them about 300 different places where the person could fit. And it was a mess. And, that's my fault. So in this version, you've got a lot more control. First of all, you're giving the tool a lot more information so that it can make better suggestions. But also, if you've actually researched the tree properly and you've got a bunch of candidates, you don't want What Are the Odds to add loads of other people in individually, all over the place. First of all, you'd rather they didn't. So I've checked the box here. Don't add unknown people, and I've said suggest people. And so it's now suggested who could have been the father. Now, in the original What Are the Odds, the person who tested was the hypothesis, and this was very confusing for people. Very, very confusing. And eventually I was ground down by having to explain it to people because it wasn't sticking after 5 years. And I made it so that you're hypothesizing as to who was the father or the mother or the parent. So that's why the hypothesis appears on the actual person. So here we've got 3 hypotheses we can see. We've got Gerald. Hypothesis 1 is that Gerald was the father, and we can see it's added a path down to David. Hypothesis 2 is that Thomas was the father, and hypothesis 3 is that Harry was the father. And we can see that Harry, with Kathy as the half sibling to the other people, is by far the strongest hypothesis at the moment. Now, if you think it might be incomplete, you can just say, sure, create unknown people. And this is an example of what it looks like when you let it create unknown people. So age-based probabilities, they're not on by default. You can say you'd like to apply the father's age. So if you check that box, you can say, all right, consider the fact that Kathy was born in 1944 and use these statistics to figure out if that has an effect on the probability. So, for example, if you've got one person who's 50 and one person who's 20, the 20-year-old is statistically more likely to have been a father, probably. And actually, specifically, using US birth data in 1944, we can apply a probability to that. So that's potentially quite powerful. Then also, if we happen to know when Kathy's mother was born, which we do in this case, we can add that information in. So we can add in that Kathy's mother was born in 1922, and then we can apply that too. And for certain years, for the US anyway, we actually have the data based on the age of the mother, how old was the father, and the inverse, which I think is very interesting, and I really wanted to get it in here, so I did. So if he checks both those boxes, we can see that we've got even stronger case for this guy, Harry Harrington, having been the father, and it says 96% likely. So I wanted to talk briefly about the scores, because as you can imagine, for a website developer, this is an absolute minefield. What if I print out 100% likely and someone goes and does something and I was wrong? It's a bit terrifying, really. Does it really mean that there's a 96% chance that Harry was the father? And the answer is, well, it doesn't really, no. 96% is the relative probability. So that means of all the hypotheses in the tree, that is the relative probability. So just based on what you've entered, that's 96% likely, with the other ones taking up the other 4%. So the issue you have is that really, for this to be valid, you have to have considered every single different possibility, which is why you might use that suggests hypotheses. And then you get a slightly lower number. Still, ultimately, this isn't something you can print out and take to a lawyer, right? You've got to do the work to figure out who's in the right place. Does everything else add up? What this tool is doing is guiding you, hopefully, towards testing someone else to prove it. So, for example, if Kathy was indeed the half-sibling of Andrew or Jennifer, could either of those be persuaded to test in order to prove it? Ultimately, these cases end with someone very, very gently asking someone if they could test, and that makes things more and more likely. In the old What Are the Odds, just to give you a contrast, this is what it looked like. And one thing that we had in the old What Are the Odds, which also confused people, was the scores. The scores were calculated the same way. They were relative scores, but I didn't try and make them percentages. I think because I worried that it would be misleading, but actually it was more confusing when they were just numbers, because 280 in one tree might be a really strong score, but in another tree that might be one of the lower scores. And it turned out that really confused people as well. So we've gone back to with actual percentage scores, which I do think, with the caveat that I just mentioned, are easier to understand. This is where we are. 96% is the relative probability. If you want to be more thorough, you're going to hit suggest hypotheses and you'll get all of the possible hypotheses in there, and you'll get a slightly more accurate figure potentially. But also remember that this is only the suggestion based on the data that you happen to have found, based on who happens to have tested. And if you have other data, it could actually completely change things. So even with these really good matches, if there were people, descendants of Thomas, who tested, actually that could change things quite a lot. So when What Are the Odds notices that there's a key sibling, in this case, who hasn't got any tested descendants, it will actually try and point that out to you, because it would be silly to jump to conclusions when you haven't really got enough data. But to give a bit of circularity to this talk, could he actually have been the man? Maybe he was in a prisoner of war camp in Japan at the time. If that was the case, then he wasn't the father. Genealogy does trump everything else. These tools can help point the way, but you always have to do the genealogy. So that is the end of the bulk of the talk. I'm going to finish just by mentioning a few other tools that I guess I would have liked to have mentioned if I had time. So we've got the coverage estimator, which was something I collaborated with Paul Woodbury and Leah Larkin on back in 2022. And that's kind of a useful way to figure out who you might want to test next if you want to get as much coverage as possible for a particular ancestor. So if you've got an ancestor where they have an unknown parent, you want to get as much of that ancestor's DNA as possible. And funnily enough, when I was preparing for this talk, I realized that my dad's got another first cousin who's quite amiable, who I have the phone number of, who hadn't tested, and I got her to test. And the test is in the post now. So that's a positive thing about both this talk and the tool. There's a tool called the Shared Centimorgan Investigator, which is very useful if you've got a set of siblings who've tested, because you can enter the segments that all of those siblings share with a mystery match, and it will kind of crunch them together and figure out how many distinct centimorgans of DNA you share with a match, and then therefore how much your parent might have shared with that match. So that's a slightly technical but very fun tool. And then finally, if you're working on inferred mapping, so that's when you figure out, I shouldn't really have got into this now, but that's when you try to figure out where a segment might have come from based on who doesn't share it. That's quite a useful segment calculation tool that I built, and you can read more on that or more about that on the blog and within the tools page. Okay, so thank you very much everyone for coming along. I hope that was coherent. If you would like to stay in touch or learn more, I've got a blog, I've got a monthly mailing list that you can sign up to, and there's also quite an active Facebook group. Those are kind of the main ways at this point. Thank you very much. So this is the coverage tool. I'll use it to just show you how you import a GEDCOM into the site, because it's the same mechanism really on all the different tools where you can do it. So just to recap, the example I always use is, say you've got a second great grandparent and you don't know who their mother was, say, or their father. You want to make sure you've got as much DNA as possible. So what you do is you click load, and then we can click import GEDCOM, get my tree up. And what it does is it loads in all of the people in that tree. And if I type Charles, it gives me all the Charleses. I can select this guy that I was talking about most of the presentation, Charles Richard Jones, and I can import his descendants. So at the moment, I haven't told the tool anyone who's tested. I've just said these are his descendants. So the tool has found all the people who are still alive and has said, oh, maybe you should test this person. So that's nice. Well, this guy I have tested, so I can put that in there. And so what does that do? It says that we've got 100% of this guy's DNA, which means we've got 50% of this guy's DNA, which means that, well, we might have 25% of this guy's DNA. This is a slightly crude tool. It uses averages, but it gives you quite a good overall sense of what your coverage might be. So overall, we might have 12.5% of Charles Richard Jones's DNA represented by just this one person. Now, there's some other people who've tested. This guy's sister, her test is in the mail now, so I can put her in. Now, that doesn't have a huge impact because obviously it just takes him up to 75%. So overall, it's taken us up to 18.8% tested. That's great. And then who else has tested? She has tested. My father has tested. So whack that in there. And his brother's also tested. Okay, that's all the testers I know about. So at the moment, we're at 32.9% coverage. Obviously, that's an artificial precision. It could be in the 20s, it could be higher, but that's a kind of guide. But I guess the most useful thing is it tells you who you might want to test next. So it wants me to test a guy called DWJ. Now, actually, I happen to know that he's no longer with us. So if I edit the details, I can just say that he is unfortunately no longer with us. Obviously, I haven't updated my tree. So that removes him from the suggestions. But it will tell me that, yeah, roughly 8.4% would be gained if I managed to test any of these people. What else can you do in this tool? If you're looking for people just who've tested at a certain place, you can say all these people are from 23andMe or they're all from Ancestry. If you've got a mistake in your tree, you can drag people around. So I can put her up here if I want to. I can make the tree vertical. So some people find the left to right thing a bit confusing. It is, but it's much more compact than this, which is the more vertical kind of descendancy view.